everyone, I'm Jessica, and I'm going to give you guys an overview of the project that I've been working on. So this project is designed around a 9th grade on-level biology class at my high school, Chattahoochee High School. The class includes a diverse group of learners, 504, IEP, and gifted. And so since this is a biology class, I am always preparing them for their end of course exam. So this lesson is designed around the GSE standard of biology for molecular genetics, which says to obtain, evaluate, and communicate information to analyze how genetic information is expressed in cells, and to construct an explanation of how the structures of DNA and RNA lead to the expression of information within the cells via the processes of replication, transcription, and translation. So essentially, I'm focusing on protein synthesis, how to go from DNA to RNA to proteins. And I have the NGSS standard as well. So the three lessons that we're going to go through show us the processes of DNA to RNA to protein, and I am focusing on an anchor phenomena of the Atlantic wolffish throughout this lesson sequence. So the first lesson is going to introduce students to the anchor phenomena, and then the third lesson is going to be them answering questions and actually constructing an explanation about that anchor phenomena now that they've learned everything at the end of the unit. And they're going to be using the BLAST database in order to answer those questions. So as I mentioned, I am going to be using an anchor phenomena. I was kind of inspired with the support of you guys, my other classmates, about your support of using an anchor phenomena. And so I'm focusing on the Atlantic wolffish, which is a fish found in the North Atlantic waters that's capable of surviving in sub-zero conditions, so in below freezing waters, which of course is a unique trait. So the students are going to be encouraged to ask questions to try to figure out, how can this be? How can the fish survive in some place that's so cold, where maybe a human cannot? And then by the end of the lesson 3, as I said, we should be able to answer that. Everything is being planned to be face-to-face because we're hoping next semester, or I guess I should say next year, in 2022, we are going to be all face-to-face. So I'm planning for that style of instruction. All right, so my first lesson is looking at the structure of DNA. But first, the students are going to be introduced to the anchor phenomena of the Atlantic wolffish. So the students are going to be asked to define problems, about their living environment. And then the students are going to start to learn about the structure and function of DNA, and they're going to do that through Kinnex modeling pieces. They're going to build a model of DNA, and that's how they're going to study the structure and function. So the science and engineering practices we're looking at are about asking questions and defining problems about that anchor phenomena, developing and using models with the Kinnex kits, and then constructing explanations and designing solutions about the structure and function of DNA. My core ideas is all about that same standard that I mentioned before, and the cross-cutting concepts are about systems and system models through the Kinnex kits, and also structure and function. How does the structure of DNA affect its function? So I was going to go ahead and show us some of the instructional materials that I've developed for this first lesson. So the first thing that we're going to do is introduce the students to the anchor phenomena. So I created a PowerPoint. That first thing you're going to see walking in the door is this interesting fish. I'm sure they're going to love the little face on the fish. So they're going to look at the Atlantic wolffish. We're going to discuss that interesting fact about how they can survive in these really cold environments. And then I'm going to give them some time to work in a pair to think about, why is this interesting? What would happen to most fish at that really cold environment? And then they're going to make a prediction, figuring out, how do we think the wolffish can survive at these conditions? So this is them asking those questions that we're going to answer at the end of lesson 3. And I added this anchor phenomena to really hopefully reinforce that kind of three-dimensional style of learning. Now, the rest of the class is going to be split between looking at the function of DNA and then building the DNA to figure out the structure through kind of an interactive guided notes. So let me show that to us. So the first bit, we're going to do kind of a mini lecture where I will be teaching the students about kind of the function of DNA, hopefully activating prior knowledge. And then they're going to be continuing on the notes on their own with a partner to use Kinnex modeling kits to build the structure of DNA and make observations and conclusions based on what they're building, kind of reinforcing that inquiry of three-dimensional learning. So these are the kits that they'd be using, the Kinnex Education DNA kits. And then here are the notes. So let's show us those interactive guided notes. So the first bit, like that first little section on function, that would be what I would be kind of working with the students to hopefully reinforce things they already know about DNA. Then we'll move into the structure. So the kids will use the Kinnex kits to build the nucleotides, and they'll kind of draw a sketch of what those look like. They'll use the 4 different bases to do so. They'll learn about the base pairs, and they will take those nucleotides and connect them together based following those rules. And I'm going to kind of, throughout this whole worksheet, encourage them to make observations. What do they notice? What do they think that means? Hopefully they'll notice that DNA is kind of opposite, right? One side's 5', the other side 3'. So they should notice that DNA has direction, and they'll make some observations. And so at the end of this, they will have a full DNA model following a sample sequence that I give them. And I can kind of use that as an assessment. And they can do a little self-check to make sure, does their model show all of those structural features of DNA? And there's a little summary and a little extension for the students as well. So this would be something that we start together as a class, but then they finish up within a partnership. All right, so again, I would use both the physical model and that note sheet as kind of a formative assessment of how they're doing. And so some modifications that I made to this lesson that I've learned about it being in this class about three-dimensional learning is I added in that anchor phenomena. I added in the modeling, and I reduced my amount of direct instruction. Typically, this would be a pretty heavy kind of lecture day, and so I really scaled that back and increased student-led learning with the modeling. Okay, so lesson 2 is looking at the full picture of protein synthesis, going from DNA to RNA to protein. So the objectives is that students will be able to construct evidence-based explanations on how DNA makes proteins. And they're going to do that by developing a model using beads of a protein that they've made with a DNA sequence. And then I'm taking it one step further where we're going to look at how does the structure of a protein affect its function. That's that cross-cutting concept we're focusing on. Well, they'll look at how could a mutation affect their protein. So our science and engineering practices, they are constructing explanations about DNA to RNA to protein. They are engaging in argument from evidence from their model, what happened with the mutation, and then developing and using models with the beads. Cross-cutting concepts and our disciplinary core ideas are the same as before, but we are now focusing on more modeling and structure and function of proteins as opposed to DNA. All right. Now, before the students get into the modeling components, I created a Keeley probe styled warm-up as a formative assessment for the students, and it follows that 3D formative assessment that we discussed in class. When we had done that formative assessment mini group project, our group really enjoyed these Keeley probes, so I decided to include one in the lesson. And so let me pull it up. So the probe is all about the production of hemoglobin in humans. Here it is. And so the Keeley probe prompt, it shows 3 different students' ideas about how DNA encodes for proteins, and the students will say who they agree with most and why, and then they'll also kind of create a visual model or diagram of how we go from DNA to RNA to proteins. All right, that's a kind of an early formative assessment. And then the bulk of the lesson will be about doing this protein synthesis bead simulation. So let's show us that. So the students are given a DNA sequence. They will transcribe and translate it. And then they're going to actually assemble the protein themselves as if they were the ribosome. And so they're going to be responsible for making their own key of which bead corresponds to which amino acid. They will fold their protein according to bonding rules that I provide. And then they're going to do a mutation, and then still with those bonding rules, see how their shape changes. And then they will kind of engage in some explanation from evidence with some analysis questions. How did that mutation affect their protein? And then structure and function. So how did the structure change perhaps the function? Do they think it would change? Why or why not? So this lesson is really designed about maybe summarizing the process of protein synthesis and would work really well perhaps after a day where the students are really given details about those transcription translation processes, and this is about kind of pulling it all together. All right, assessments, of course, is that probe and the activity. And so how did I make this more three-dimensional friendly? I added that probe as a grade assessment. And then I added a little bit more of a focus on the structure and function, that cross-cutting concept. All right, and then lesson 3. Lesson 3 would be at the very end of this unit, and it's all about answering that anchor phenomena about the Atlantic wolffish. So the students are going to be actually given a DNA sequence from the wolffish. They are going to transcribe and translate it. And then they're going to use the BLAST database to figure out what protein that actually codes for. And hopefully that protein should answer our question as to how the wolffish survives. So we are going to be analyzing and interpreting data, obtaining, evaluating, and communicating information. All of that's coming from that BLAST database. Then we're going to take that info and construct an explanation to answer those original questions we had. We're still focusing on the same main standard, but this time our cross-cutting concepts is still structure and function. So how does maybe the structure of the protein affect the function of the fish? And also cause and effect. So how does maybe that DNA affect how they can survive? So this whole lesson is all about this BLAST activity. So I'm going to first start off in this lesson by introducing the students, reminding them of the phenomena, and then they're going to work in partners or small groups on the actual activity. So I'm going to show us what that looks like right here. All right. So the first bit goes over the instructions on how to use the database. It can be tricky, so that's why I'm going to spend some time working with the students on that. Here is the wolffish DNA, transcribe and translate. They will figure out the protein. It will describe the function to them. They'll put it in their own words. Now, the answer to it is that that fish does have a protein that's kind of like an anti-freeze protein, so it prevents them from freezing. So that will be our solution to our mystery. The students in their analysis section will go over that. So they will make a claim answering their questions. They will justify it with the evidence that they got. They will explain kind of how does DNA encode for proteins, bringing it all together. Some conclusion about the BLAST program and application. And then as an extension, I give them, they could pick from one more of these DNA sequences from a different organism and figure out the protein, just as another fun example. Okay, so once they get to the end, they are finished, and they should hopefully have all of the answers to how to the anchor phenomena, how the wolffish can survive. So I'll use this as a main formative assessment, probably right before we take our summative assessment, to try to really make sure they understand all the big ideas. And then how I try to make this more three-dimensional friendly is I pretty much created this activity as a way to answer that anchor phenomena that I had implemented at the start of my lesson. So I hope that you guys enjoyed that special guest. That was my cat, sorry. But I hope that we enjoyed taking a look at my project here, and I can't wait to look at yours. So bye, guys.